Microbiomix is a metagenomic analysis of a patient's entire microbiome. We report nearly every organism that lives in the gut, as well as the microbiome's genetic potential to produce certain metabolites. These metabolites have both beneficial and harmful effects on health. There are several pages of the report containing a large amount of information about a patient's microbiome. We've organized the report into sections that make it easy to quickly summarize this important information. There is some repetition throughout the report, starting with basic summary information, then moving into more detailed pages. The report summary is located at the beginning of every report. This is where you will find clinically actionable therapeutic considerations that will help improve a patient's gut microbiome. Here we see that the patient has 2 organisms that are considered pathobionts or potential pathogens. These may cause diarrhea, and the clinician should address this according to the patient's clinical picture. The end of the report lists every single organism present, including these 2, so you can visit that section to learn more about the organisms. Other recommendations are listed here as well, and we will look at those more in depth in a later section. For the busy clinician, the report summary contains all the actionable information they would need in order to come up with a treatment plan for a patient, without necessarily having to visit the rest of the report. The next section of the report is called Your Report Overview, and the first graphic we see is the Health Indicator Score, which uses a data-trained algorithm that distinguishes healthy from unhealthy patterns. It shows how well the patient's microbiome compares to a healthy cohort. A higher score is ideal. A lower score suggests that some work may need to be done to optimize the microbiome. All the therapeutic considerations we just saw on the previous slide can help move towards a healthier microbiome. Next in the report overview is the microbial diversity measurement. Diversity assesses the number of different microorganisms, or richness, and abundance of each, or evenness. A higher diversity is ideal. If diversity is low, a diet that includes a variety of plants as well as fermented foods may be recommended to encourage microbiome diversity. This measurement is also known as the Shannon Index, which is widely used in scientific research. Our clinical reference guide goes through many of the markers we measure on this profile and can be found on the Microbiomix page under Educational Materials. The guide discusses the clinical relevance and therapeutic considerations for each marker. Next in the report overview is a quick snapshot of the 5 most abundant species. Here we see the question, does your sample have an overly high abundance of any single organism? In order for that to be a yes, an organism's abundance has to be greater than 10%. In this example, we see one organism that is greater than 10%. This relates back to the topic of diversity that we just talked about. In general, it is beneficial if a single bacteria does not dominate, and eating a varied diet helps with diversity. If interested, you can read more about these individual bacteria at the end of the report. The next section is called Your Key Insights and is a high-level description of important metabolites produced by the microbiome. Remember, we are not measuring the metabolites themselves, but rather the microbiome's genetic potential to produce them. There's a high-level description of the metabolite and commentary telling you if your level is good or not. The bottom section gives more detail about the metabolite and ways to influence levels with diet. The evidence rating at the bottom shows how much scientific literature there is to support this marker, with 5 stars being a robust amount of evidence. In this example, we are looking at the metabolite butyrate, which is an important fuel source for intestinal cells. The potential to produce butyrate is not at a good level. There are suggestions at the bottom for foods rich in resistant starches, such as lentils, peas, beans, and rolled oats, to encourage the microbes to produce butyrate. We are looking at the genetic or genotypic potential of organisms to produce either beneficial or harmful metabolites. We are not measuring the metabolites themselves, which would indicate the phenotypic expression. Although we do have testing to directly measure some of these metabolites, such as our GIFX comprehensive stool profile. The good news is that we can influence the microbial genome composition and expression with diet and lifestyle interventions to improve overall health. The next several pages continue through the key insights of other metabolites. The key insight pages are helpful for both patients and clinicians to understand. The metabolites reviewed in the key insights are also included in the next section, the Digging Deeper section, for those who are seeking more information. Some clinicians choose to focus on the key insight section versus the Digging Deeper section. The Digging Deeper section gives a summary of all of the assessed metabolites, whereas the key insights focuses on select metabolites. We will go through the Digging Deeper section in detail so you can become familiar with all of the metabolites assessed on the Microbiomix report. The first chart we see here describes the DNA sample composition compared to a healthy cohort. The microorganisms in the gut consist mainly of bacteria. We also assess other classes of organisms, including archaea, eukaryota, which includes fungi and parasites, a novel category, which is DNA that cannot yet be identified, and human DNA. If there is greater than 4% human DNA, this could indicate gut inflammation, and further testing may be warranted. Inflammatory markers on the GIFX can help to identify if there is inflammation. Human DNA can also come from accidentally touching the swab during collection. The next section, Microbiome Digestion Potential, looks at how efficient the organisms are at breaking down fuel sources, including fiber, mucin, and protein. Adequate fiber intake actually plays an important role in all three of these measurements. Next, we look at each of the microbial metabolites assessed on this report. Here's a list of the microbial metabolites assessed on the Microbiomix report. You can see that there are diverse clinical implications, including cardiovascular risk, SIBO or small intestine bacterial overgrowth, kidney stone formation, and vitamin production, to name a few. Now let's go into each metabolite in detail. Again, our clinical reference guide found on our website can be helpful while reviewing these markers. Hexaacylated LPS, or hexa-LPS, is a pro-inflammatory component of certain bacteria and is associated with many diseases, including obesity, heart disease, and diabetes. Saturated fat helps to shuttle LPS across the intestinal barrier, so decreasing saturated fat can help reduce LPS from entering the bloodstream. In this example, this would be a good recommendation since the finding shows a high potential for the microbiome to produce LPS. The recommendation to reduce saturated fat can be found at the very beginning of the report, in the report summary section, which we already briefly discussed. This marker is also discussed in the Key Insights section describing the microbiome's potential to negatively impact the gut through inflammation. Additionally, omega-3 fatty acids have been shown to reduce plasma LPS levels. Some metabolites will have a plus or minus sign next to it, indicating whether the metabolite has beneficial or harmful effects on health. Here we see a minus sign since there are many diseases associated with the marker. The percentage is the relative abundance of all organisms in the sample able to do that function. In this case, 2.17% is slightly higher than what you would find in a healthy cohort. Finally, there are hyperlinked references at the bottom if you're interested in digging deeper into the literature. Methane gas is associated with slower transit or constipation. Certain organisms are capable of producing this gas, but in this example, the potential to produce methane is not detected, or ND. We do offer a SIBO breath test that assesses methane gas. Trimethylamine, or TMA, can be transported to the liver and converted into a substance called TMAO. TMAO is associated with cardiovascular disease, cancer, and diabetes. Compounds from cruciferous vegetables can help to reduce this conversion to TMAO. Additionally, red meat contains carnitine, which can be converted to TMAO. This example shows a high potential to make TMA. Excess ammonia production has been observed in individuals with impaired gut barrier function and gut inflammation. This example shows a low potential for bacterial ammonia production, which is good. You can refer to the clinical reference guide for therapeutic considerations when it's high. Since ammonia comes from protein, ensure that protein intake is balanced with enough fiber and resistant starch intake. Remember, any abnormal findings in this section will show up as recommendations at the very beginning of the report in the report summary section. Some strains of the bacteria Bacteroides fragilis can produce a toxin that may cause diarrhea and intestinal inflammation in some people, while others may not experience any symptoms. In the example above, the potential to produce the metabolite is not detected in the microbiome. If it is detected and the patient is experiencing diarrhea and other causes have been ruled out, this may require treatment. It's important to correlate with patient symptoms. Beta-glucuronidase is an enzyme associated with potential recycling of toxic compounds, including estrogen and other hormones, medications, and environmental toxins. Glucomannan is a prebiotic fiber in konjac root that reduces fecal beta-glucuronidase activity. The potential to produce beta-glucuronidase is low, and this is a good finding. We measure beta-glucuronidase directly on our GIFX comprehensive stool profile. Hydrogen sulfide gas is responsible for the rotten egg smell of flatulence. High levels are associated with inflammatory bowel disease and colorectal cancer and can disrupt the gut mucus barrier. Foods high in resistant starch and FOS, or fructooligosaccharides, can reduce hydrogen sulfide production and may be beneficial in this example since the production potential is average. Low potential to produce hydrogen sulfide is ideal. High levels of circulating branched-chain amino acids are associated with obesity and type 2 diabetes. Physical activity helps the muscles to use the branched-chain amino acids. Additionally, increasing fiber can decrease microbial branched-chain amino acid production and affect amino acid metabolism. In this case, the potential to make branched-chain amino acids is low, which is good. Genova's NutriVal assesses plasma branched-chain amino acids. Oxalates from our diet can become calcium oxalate kidney stones if they are not metabolized properly by the microbiome. Here, an ideal finding is an average to high potential to break down oxalate, so in this example, the patient is in a good range. If the potential is low and the patient does not have kidney stones, this may not be an issue. If the patient does have kidney stones, a low oxalate diet may be beneficial. Our NutriVal or Metabolomix Plus profiles assess metabolites within the glyoxylate pathway, including oxalic acid. We look at two markers on this report related to GABA. The bacteria can both produce and consume GABA, which is a calming neurotransmitter. Low levels of GABA are associated with depression and anxiety. In general, a balance between production and consumption is preferred. In this case, we see slightly more GABA being consumed versus produced. The role of the microbiome and associated treatment is still being studied. If the patient does experience a mood disorder, it's important to seek professional help. Three-indole propionic acid, or IPA, is a beneficial tryptophan metabolite. It's a strong antioxidant that can protect the nervous system from damage and may help prevent type 2 diabetes, suppress inflammation, and maintain the gut barrier. In this case, the potential to produce this metabolite is average, which is good. If it's low, high-fiber foods including whole grain wheat and rye, as well as ellagic acid from foods like pomegranates can help. Histamine is produced both by gut bacteria and human cells. Gut bacteria that produce histamine may be higher in patients with asthma. People with food allergies and IBS may be more sensitive to histamine produced in the gut. In this case, the microbiome potential to produce histamine is high. Strategies to reduce overall histamine load may be considered. Outcome studies are lacking regarding microbiome modulation to alter systemic histamine levels. Butyrate, propionate, and acetate are short-chain fatty acids produced by bacterial fermentation of fiber or lactate. Short-chain fatty acids are beneficial and have a range of activities important for gut and overall health. Short-chain fatty acids are measured directly on the GIFX comprehensive stool profile. An average to high potential to produce these is ideal and can be seen in the example here with the exception of butyrate, which is low. If any of these are low, there are suggestions for specific prebiotic fibers or resistant starch to increase levels. The microbiota produce many vitamins to nourish both the human as well as themselves. They can produce vitamin B12, folate, biotin, B2, and vitamin K. Of all the vitamins assessed on this profile, folate contributes the most to human needs for this vitamin. If the microbiome potential to produce any of these vitamins is low, it's important to ensure adequate dietary intake. The next section of the report looks at species of interest. These species tend to have more literature support and clinical relevance. The detected species include more detail at the end of the report if you're interested in learning more. First, we look at bacteria, and the list shows whether it was detected or not detected. Also, the same plus and minus signs are present that we've seen throughout the report, indicating whether the organism is beneficial or detrimental to health. We also assess archaea, as well as yeast and parasites. Finally, we come to the last section of the report, the microbial profile. This section includes every identifiable organism housed in the patient's microbiome. Your microbiome profile, broken down by phylum, shows the abundance of each phylum relative to a healthy cohort. It's a quick snapshot of how the patient compares to others and how balanced or imbalanced they are. The remaining pages go into much detail about each organism present. It shows the abundance level, and here we can see that this particular bacteria is high. You can learn about its fuel sources, metabolites produced and consumed, as well as any emerging research. We already discussed all of these metabolites in the previous section. We don't encourage getting caught up in the details of every single organism on this report. This information is here for reference if needed. Ultimately, the first few pages of the report outline the best course of action clinically in the report summary section. We did see earlier in the report review section that there were 2 potential pathogens present, and this section can give more information about those. The organisms are not in alphabetical order, but in order of abundance. So you can use your search function, such as Control F, to find a specific organism. The presence of a potential pathogen or pathobiont does not automatically necessitate treatment. In many cases, these organisms are still being studied and known treatments are not available. It's important to look at these in the context of the clinical picture. It is possible for a patient to have these pathobionts and be asymptomatic. In review, we revisit the report summary located at the beginning of the report. We just discussed the pathobionts. This patient has high human DNA, which would mean there is gut inflammation, which can be checked with stool inflammatory markers such as calprotectin, eosinophil protein X, or secretory IgA. Additionally, it's important to make sure the patient didn't come in contact with the tip of the swab during collection. They also have an elevated potential to produce hexa-LPS, so reducing saturated fat may be beneficial. The potential to produce TMA is high, so evaluating red meat intake and increasing cruciferous vegetables would be important. Butyrate production can be stimulated by consuming foods high in resistant starch or pectin, and examples are listed here. GABA consumption is high, so if the patient struggles with mood disorders, it's important to seek professional help. The potential to produce histamine is high, so it's important to reduce the overall histamine load. And finally, folate production potential is low, so it's important to eat high folate foods such as green leafy veggies, legumes, and nuts, and determine if folate supplementation is necessary. Thank you for joining us today.